So I will confine today's lecture on structure, functions, and secretions of liver and gallbladder. So what is liver? Liver is the largest organ in our body. It weighs about 1.4 to 1.6 kilogram, and in women it is relatively less, 1.2 to 1.4 kilograms, and its main function is metabolic. It has synthetic as well as excretory functions. It has, normally our organs are having blood supply, single blood supply, means from artery. But in liver, there is dual supply. Blood is coming to liver from two sources. One is from hepatic artery, another is from portal vein. This portal vein is collecting blood from GI tract and spleen. Okay, so these are the dual source, but coming out from the liver by the hepatic vein it drains the blood of liver that is coming out of liver, will pass through hepatic vein, and then it will enter into IVC, inferior vena cava, and that will drain its blood into right atrium. Okay, so this is the circulation. And liver is extremely resilient. It means if you try to deform it, again it will come to its normal shape. So that's why it is resilient. When we suspect liver disease, particularly cirrhosis, in such condition there is stiffening. Liver cells are replaced by fibrotic cells, fibrous cells. As a result, it becomes stiff. So it loses resilience. So to study this, what happens? We apply force into the liver, it is deformed, and after the application, after the withdrawal of the force, normally again the liver comes to its original position. That means it is having elastic nature, elasticity. That's why one test is performed, that is elastography and fibroscan. So by this method, it is ultrasonography, one kind of ultrasonography, shear wave or ultrasound wave is transmitted to the liver, so it creates pressure and then withdrawn. So with this, how much liver is compressed and how much it came back to the normal position. So from that we determine how much liver has been lost or it is fibrosed. Okay. So this is important. Another important thing is, in liver, it's regenerate very fast. If some portion of the liver is removed, then that part is again regenerated. It takes near about 2 months. Okay, so that is one of the important aspects. And liver is having a lot of bile ducts. Because one of the important function of the liver is secretion of bile. So these biles are coming through bile ducts. So from different lobes, it has got 4 lobes, you know that? Liver has got 4 lobes. So right lobe, left lobe, caudate lobe will be coming into. So different lobes are there. So from all these lobes, either it is from quadrate lobe or left lobe or right lobe, from all the lobes bile ducts will be coming, they will be joining together and will form right hepatic duct, left hepatic duct. Both will join together and form a common hepatic duct. Now, from this common hepatic duct, another duct will be entering into gallbladder. So that duct, initial part of that duct is cystic duct, then gallbladder. Okay, and after that, that is cystic duct, hepatic duct, they will join together, will form common bile duct, CBD, common bile duct, that will enter into the duodenum through ampulla of water. Okay, so this we'll discuss later. And macroscopically, the liver is divided by fossa on its surface into two halves, right and left lobe, right lobe and left lobe, and it is connected to the diaphragm and abdominal wall by different ligaments. What are these? The falciform ligament, and that will also divide the right and left lobe, and the coronary ligament, right and left triangular ligaments, and the fibrous round ligaments. So these are the different ligaments, 5 ligaments by which liver is connected to, attached to the diaphragm and also the abdominal wall. And microscopically, if we study, take the sections of liver and study under microscope, we will find each lobe is made up of thousands of lobules, hepatic lobules. See how they look like. These are the hepatic lobules, hexagonal shape. At the center, there will be a cavity. This is central vein. Come in. Come in. Where's your leg? At the center, there is what? There is what? Central vein. From all these lobules, from all this, see, at the corner, these corners, you will find some vessels, cross section of vessels. 3 different vessels are there. 3 different vessels are there in each corner. And because of these 3 vessels, they are called triads. This corner is called triads. And these 3 vessels are branch of hepatic artery, branch of portal vein, and branch of bile duct. So what you will get is at the triads? Hello? What you will get at the triads?
The 3, one artery, a vein, and section of bile.
Right. Okay. Now see, what are this? How you will identify? Say you have to, you have spotting, no? So how you will identify this is a section of liver? You will get the lobules, you will get the central vein at the center, and radiating columns of hepatocytes, like the spokes of bullock cart. There is wheel, in the wheel there are spokes. So that way the hepatic cells are arranged like radiating columns. Okay? So here I can see that radiating columns, isn't it? So they are arranged like this. More minutely we will see. This is one triad. This is one tract. What is this? This is the bile duct. Sections of bile duct. And this is sections of portal vein. And this is section of artery. Arteriole. So see, one of the important thing, blood is coming through artery, also through portal vein. Both this blood, portal blood and arterial blood, they are mixing here. Both these blood are mixed here. What are these? Sinusoids. These tubular structures are sinusoids. Sinusoids are in between. This is one radiating column. This is another radiating column. In between, there is a tubular structure. So this blood is drained here. So arterial blood is drained here. Also, what is your apron? So where the blood, arterial blood and portal blood will be mixed? In the liver sinusoids. What are sinusoids? The space in between radiating column of hepatocytes. Now the blood is coming through these two vessels, and where this blood will go? They will enter into the central vein. So blood is coming to artery, entering into the hepatic lobule. Blood is coming from portal vein, entering into the sinusoids. Here it is mixed. Then the blood is entering into central vein. Now all these central vein joined together will form from the different lobes. They will be joining together and will form hepatic vein. Now this hepatic vein will join inferior vena cava. From inferior vena cava, this blood will be coming to right atrium, to the heart. And again it will be circulated. So first, whatever we eat, after digestion, it enters first into liver through portal vein. This portal vein also carries blood from another organ, spleen. This portal vein also collects from mesentery. We have superior mesentery vein, inferior mesentery vein. So they join into the portal vein. Okay, so actually portal vein is collecting blood from which organs? Which organs? GI tract and mesentery. Okay, so this blood is drained into the liver first, and because why liver first? Why liver first? Because liver has some storage function. So whatever is in excess, it will store there. And during need, it will supply those products. And from GI tract, some unwanted material may come enter into the blood. So that has to be detoxified. So liver has some cells, Kupffer cells. They are phagocytic in nature. Their location is in the sinusoids. So blood is entering into the sinusoids. So now in the sinusoid there are free Kupffer cells. Now this Kupffer cells engulfs the organisms and toxic substances and destroys it. So then after that, okay, now the blood there is no problem, no organism, now it can be circulated into the general circulation. Okay, So you understood this? More detail. What are these? Radiating columns of hepatocytes. What are the cells of liver? Name of the cells? What is the name of the liver cell? Hepatocyte. Hepatocyte. So these are hepatocyte. 98%, 97% of the cell constituent is hepatocytes of liver. Okay. So these are what? Kupffer cells. What is their function? Protective function. They engulf, destroys microorganism toxins. Now, these cells, hepatocytes, they actually synthesize bile. They produce a secretion, a juice that is called bile. Now, this hepatocytes makes this bile. These are the hepatocytes. So this hepatocyte makes this bile and secretes this bile into a fine tubular structure, canaliculi. So this hepatocyte, after synthesizing this bile, secrete it into this canaliculi. Now all these canaliculi join together. All these canaliculi join together to form a bile duct. Now this is the triad. A triad, there is one section of bile duct, that is the bile duct. So these cells are making bile, secreting into the canaliculi. Now this canaliculi draining this bile into the bile ducts. Okay? And here, this is the sinusoids, this is the hepatocytes, these are the hepatocytes. So blood is entering here, mixing here. So these hepatocytes, whatever is required, it collects from this blood. And whatever it has to secrete, say this hepatocyte has stored glycogen. It has stored glycogen. Now blood sugar level has become low. So sugar level has to be increased. The hepatocyte has storage of glycogen. So it will break down this glycogen into glucose. Now this glucose will be liberated from here into here. Then what is this space called? This space is called space of Disse. Space of Disse. So this will be liberated into the space of Disse, and from that space of Disse, this will enter into the blood. Now this will be circulated. Similarly, liver cells can store iron, copper, vitamins, A, D, E, K, all these fat soluble vitamins, liver hepatocytes can store. So if I need these vitamins, they will secrete it from the hepatocyte into space of Disse, and from the Disse it will be liberated into the sinusoid blood in the sinusoid, and from here it will be entering into a central vein. This central vein, so all this blood will be drained here, all this from sinusoids, all the blood will be drained here. Central vein, this will again join to IBC, inferior vena cava. Has you got the idea? Got the idea? Okay, now tell me. Today I ate rice. After digestion, what will be the product? After digestion, what will be the product? Glucose. Now, from the GIT, from the lumen of the gastrointestine, where this glucose will go? From lumen, where it will go? First, from the lumen, where it will enter? Where it will enter? It is the lumen. So it has to ultimately come to blood. How it will come? There are the mucosal cells. So first it has to enter within the mucosal cells. But it is not so. It's a simple diffusion, it cannot enter. It needs active transportation by the glucose. There it has got transporter, SGLT1. Through SGLT1 it will enter with the help of SGLT1. It will help. So it will first enter into the enterocyte. From enterocyte it will enter into the capillaries of water portal vein. Water portal vein. First make it clear. Water portal vein. We are pronouncing portal vein, pronouncing repeatedly. Number of times this word is coming. Water portal vein. Any idea? None.
The vein that carries deoxygenated blood from the liver.
Suppose this is an organ. So this organ is having multiple cells. Okay. So they need nutrients, they need oxygen. They will produce something that has to be secreted into the blood. So there should be blood supply. Who will supply? Artery, the branches of artery. So one branch of artery will come here and it will form capillaries. It will form capillaries. These capillaries are arterial end of the capillaries. Now this from capillaries okay. Arterial branched into capillaries, arterial end. From this arterial end, venous capillaries is formed. They are joining together, will be forming venule. Now this venule will join together, will form veins. So one organ is getting blood. So one artery, suppose it will be supplying spleen. It will be supplying blood to skin or any other organ, or my thyroid gland. So one artery will be coming here. It will form branches and it will form capillaries. From these capillaries is again joined together. It will be venule will be formed. It will carry it away. So one source, one input, one output. So artery, they form capillaries, and from the capillaries vein is formed, and this is drained into the heart finally. But there are some organs where we will find some organs, will find what? Some blood vessels, they originate from capillaries and again end into capillaries. There are two, at the two ends there are formation of capillaries. They get originated through the formation of through union of capillaries, then again they will be ending through the branching of capillaries, through capillaries into the organ. So two times, capillary formation is there two times, but in all other cases there will be single time capillary formation. So in vein where you will get capillaries at the both end, then these blood vessels, these veins will be called portal vein. Similar portal vein you will be getting in between hypothalamus and pituitary gland. This is called hypothalamico-hypophyseal. Hypothalamico-hypophyseal. Hypophysis means pituitary. Hypothalamico-hypophyseal portal system. And what is the function of this portal system? Hypothalamico-hypophyseal. We know the functions of the anterior pituitary and intermediate pituitary are controlled by hypothalamus. Hypothalamus is controlling the functions of anterior pituitary and intermediate pituitary. Okay, how they are controlling? They are forming some releasing factors, or they are secreting some inhibitory factors. So with this releasing factor and controlling factor, they control the functions of cells of anterior pituitary. Okay, so they will make balance. So these hormones secreted from pituitary has to come to the pituitary. How this will be coming? This will be coming through this portal hypothalamus. We release these releasing factors or inhibitory factors into the portal vein. Now this will be drained into pituitary. Now if GRF or TRF is released, now this TRF will cause TSS production. Now this TSS from pituitary will be going to the thyroid gland, and it will stimulate the thyroid gland for the liberation of T3 and T4. Okay, so that's why it is important. How this TRF will come from here to, okay sir, it can come through another loop. What is this loop? Because hypothalamus is getting blood through some arterial sources, and it has got some veins. So these veins will be coming through, blood will be from hypothalamus, will be coming, suppose there is no portal vein, so it will be coming to heart, then this TRF will be distributed throughout the body, it will be mixed with 5 liters, 6 liters of blood. So their amount will become, ultimately concentration will become very less. But here if I drain the blood, then this product directly from hypothalamus to the pituitary, then its concentration will be much more. So that's why this has been created. Similarly, can you name some hormones of the posterior pituitary? What? Huh? One is vasopressin, another is oxytocin. Posterior pituitary synthesize this or they release this? Confusion. Whether posterior pituitary synthesize these two hormones or they release it, just. No, they don't synthesize. Posterior pituitary cannot synthesize oxytocin and vasopressin. Now where this will be synthesized? Again it is in hypothalamus. Supraoptic and paraventricular nucleus, these are the sources. Now question is, how they will come to the posterior pituitary? And these two are neurosecretions. These two are neurosecretion practically. Should they come, these are synthesized in the supraoptic and paraventricular nucleus. Means there are some cell bodies. A group of cell bodies are there. These cell bodies of a neuron, they are actually manufacturing it. Now they will be passing through the axon. And the terminal of these neurons, terminal is in posterior pituitary. The terminal is in axon. So it is synthesized here. When these are stimulated for release, so they will be released from the nerve endings like acetylcholine or adrenaline. So they will be released in the posterior pituitary. From posterior pituitary, when it is released from the nerve endings, they will enter into the blood. Then it will be circulated. So what is this tract? These neurons coming from hypothalamus to posterior pituitary. Hypothalamico-hypophyseal tract, not vein. Understood? So, keep it in your mind. Anterior pituitary cells are stimulated by hypothalamus. Hypothalamus is also an endocrine organ because it is producing some releasing factor, inhibiting factor. With this, releasing factor will stimulate, inhibiting factor will inhibit. So with this, like sympathetic parasympathetic reaction, so the hypothalamus controls the pituitary by releasing factor and inhibiting factor. So with this controlling mechanism, they actually control the functions of cells of the anterior pituitary. So how this will be coming? Through portal vein. What is this portal vein? Hypothalamico-hypophyseal question comes. And how the hormones of posterior pituitary comes from hypothalamus to posterior pituitary through a nerve tract. What is this nerve tract? Hypothalamico-hypophyseal nerve tract. Is that clear now? Okay, good. So now we could understand. So what is portal vein? So they will be having capillaries at their both ends. But in other case, in other organs you will find only one time capillary formation will be there. Artery is coming, arteriole is coming, it will produce capillary. From this capillary, vein will be originated. Ultimately, no more further formation of capillaries. It will then ultimately will come to the heart. Okay, so let us study the functions of liver. One of the most vital function of the liver is metabolic functions, because it is the central part of the metabolic, all the metabolic processes are taking place here, and the center, or here you will find all the metabolic processes are actually happening in liver. So let's see what are these So one is main function is metabolic function, we will discuss, and the second is storage function, and the third is excretory or secretory functions. Then we will find protective function, then we will find circulatory function. This is portal circulation, which we will discuss. And then coagulation function, we know all this coagulation not all, most of the coagulation factors are synthesized by fibrinogen. Where from it? Without fibrinogen, is it possible coagulation? Not possible. So from where it is synthesized? Liver. Prothrombin. Where from it is synthesized? Liver. And the blood coagulation factors. Factor 5, factor 7, factor not 9, factor 10, factor 11. These are also synthesized from the liver. So and thereby it helps in coagulation. Okay. And synthetic function. So we'll see, or endocrine function, synthetic function we'll discuss one by one. This is the metabolic function. What is this? It participates in carbohydrate metabolism. We know gluconeogenesis. Where it takes place? In liver. When there is shortage of sugar in your blood, liver comes forward to rescue. Okay, without sugar you cannot survive. Without glucose, how you generate your energy? So, it may happen because you may not get food for some days, or you have undergone starvation, fasting. So then what to do? So liver initially, it knows. So in future, this condition may happen. So that's why it stores some carbohydrate in the form of glycogen. So when there is excess glucose, when there is excess income, we deposit in bank. When we will be needing, we will draw the amount and we will disburse. Similarly, when there is excess glucose, liver takes up this excess glucose, converts it into glycogen. When you will be needing, you are in crisis, this glycogen will be broken down into glucose by the process of glycogenolysis. Glycogen breakdown, glycogenolysis. It happens only in liver. Though, one important question. Muscle can also deposit glucose as glycogen, but it cannot produce free glucose. Why? Because one enzyme is lacking in muscle. And what is this? Glucose 6-phosphatase. And because of this, enzyme is absent in, though glycogen is present. Actually, no need of glycogenolysis in muscle. Because why there is no? It is the need of the muscles, they actually need glucose. So muscles are having storage for future use. You may say, sir, blood sugar level has dropped. So this glycogen can be broken down and it may form glucose and liberate it into blood. So blood sugar level may rise. But ultimately, why to raise the blood sugar level? To supply oxygen and glucose to the muscles. So what is their use? No need of supplying, breaking this glycogen into this. Then what muscle will do? Muscle, suppose no glucose is supplied through blood, so it will use its own glycogen. So how it will? There will be glycogenolysis. Glycogenolysis means there will not be production of free glucose. There will be glucose 6-phosphate, glucose 1-phosphate, then glucose 6-phosphate, then glycolysis. In glycolysis, what happens? Glucose is first converted into glucose 6-phosphate. So from glycogenolysis, in muscle, it will be converted into glucose 1-phosphate, then it will be converted to glucose 6-phosphate, then rest part is like glycolysis. Okay, so in such case, when there is decreased blood sugar level, liver immediately undergo glycogenolysis and it will liberate glucose from glycogen. So liver cells, it is having glycogen. It is formed from glucose when blood sugar level was high. So at that time it took up glucose and converted into glycogen by the process of what? Glycogenesis. See, one of the function is glycogenesis. So by the process of glycogenesis, it will convert glucose into glycogen. Now when blood sugar level came down in blood, then what will this will be broken down into glucose. How? This was glycogenesis. It is glycogenolysis. So liver has another mechanism by which it can increase the blood sugar level. How? What is this? What is gluconeogenesis? Production of synthesis of glucose from non-carbohydrate substance. What are these? Glucogenic amino acids, glycerol. From where you will get glycerol? After digestion of lipid. Triglyceride means glycerol and fatty acids. So this glycerol is absorbed into the blood. From the blood, the liver will get this glycerol. Okay. So from this glycerol, the liver can use for the production of glucose by the process of gluconeogenesis. Similarly, from lactic acid, also glucose can be formed. So non-carbohydrate substance from the glucogenic amino acids. Some amino acids can be converted into glucose by this. Not all the amino acids. Those who are glucogenic, who can produce this, they are called glucogenic amino acids. So what happens after deamination? See, I uttered the term deamination. What is deamination? Amino acid has amino group. There is one enzyme called deaminase. So deaminase can take away this amino group from the amino acid in the form of ammonia. Okay, let's see. You have taken a lot of protein. So what will be there? It will be digested into different kinds of amino acids. And we actually require the essential amino acids. Because all other amino acids I can synthesize. So no need. All these amino acids, I went to a restaurant and they are all into a marriage party. So there I ate lot of proteins. Actually, I don't need this much of protein. So there are a lot of proteins. I cannot, like glycogen or fat, it cannot be stored. Protein cannot be stored for future use. So what will be? They will be catabolized. So these are catabolized first. First it will undergo deamination. So from amino acid, ammonia group will be liberated as ammonia. Now this ammonia is toxic. When it will combine with water, what will be formed? Ammonium hydroxide. What is ammonia? It is acidic or base, alkali? So there will be ammonia toxicity. So what can be done? liver comes into action. Okay, I will convert this ammonia into less toxic product, which is urea. It has got a cycle by which it converts ammonia into urea. What is this cycle? Ornithine cycle. Okay? Or it is urea cycle. So it detoxifies. Why I have produced ammonia? For deamination of these amino acids. Why I deaminated it? Maybe this amino acid, after deamination, this will be required for synthesis of some other compound. Or I may require this amino acid for production of glucose by the process of gluconeogenesis. That's why it has undergone deamination. And liver has another function. It converts one kind of amino acid into another kind of amino acid. It transforms one amino acid into another. Okay, how? By either deamination or by transamination. Transamination means transferring amino group from one amino acid to another compound, another amino acid. So transferring this, not removing as ammonia. Transferring the amino group from one compound to another. So that is called transamination. And deamination is removal of amino group as ammonia. It all takes place in liver. Understood? So, another thing, you have learned alternative pathway of glucose metabolism. What is it? HMP shunt. Hexose monophosphate. This is also called pentose phosphate pathway. You are not eating any pentoses. You are eating hexoses, polysaccharides. But your DNA, RNA requires pentoses. How this will be generated? You are not eating. So it should be produced in the liver by pentose phosphate pathway. Here hexoses are converted into pentoses. So these pentoses may be required by the dividing cells for DNA, RNA formations. Okay? So in this way, liver takes part in carbohydrate metabolism. Next, coming to hormone metabolism. Hormone metabolism, okay, I'll come later. Then fat metabolism. Beta oxidation, you know. Beta oxidation takes place in liver, also in muscles. Okay? And there is synthesis of fatty acids. See, you are eating what? You are lot of carbohydrate daily eating. You are becoming fat. How? Eating carbohydrate, producing fat. That means carbohydrate can be converted into fat. In carbohydrate metabolism, what you are producing in between during metabolism, acetyl coenzyme, isn't it? What is the end product of glycolysis? Pyruvic acid. Then what will happen to it before entering mitochondria? Before entering mitochondria TCA cycle, it has to enter into the TCA cycle, no? Glucose in blood. This is a muscle cell. So from here, it has entered here. Purpose, energy. This is mitochondria and this is cytoplasm. This is a cell. Nucleus, cytoplasm, cell membrane. So this from blood it will enter into the cytoplasm. Purpose is to generate ATP. So first it will undergo glycolysis. After glycolysis, if oxygen is sufficient, oxygen is there, condition is aerobic, then end product will be pyruvic acid. So here will be what? There will be glycolysis. So in the cell, there will be glycolysis. Of what? Of glucose. Glucose will undergo glycolysis and it will produce pyruvic acid. Now this pyruvic acid will enter into the mitochondria for further oxidation, because only minimum amount, glucose is having lot of energy, only minimum portion is only by glycolysis, only a minimum portion of ATP I got. The maximum amount is retained within pyruvic acid. Now this pyruvic acid, before entering here, it will be converted into acetyl CoA. Acetyl coenzyme A. Now this will enter into the mitochondria. Here it enter into join in the Krebs cycle, in the TCA cycle. There it will be oxidized, and 12 molecules of ATP will be produced from each acetyl coenzyme A. So ultimately, during this process, you will be getting ATP or GTP at substrate level, and you will be getting NADH2, FADH2. This NADH2, FADH2 will be oxidized in the electron transport chain. From each NADH2 you will get 3 ATP, from each FADH2 you will get 2 ATP. So in this way, ultimately we get ATPs. So you should know the roots.